Everyone is doing well. This is week 3, chapter 2 lecture. So just some general announcements here. So this is going to be, chapter 2 is a lot. There's a lot of content in here that you're going to be responsible for on the exam. This will be, probably one of the longer lectures this semester. That's my guess. This entire chapter basically summarizes a majority of the information that you'll cover in Bio 1 cellular processes. So there's a lot of content here. Now, just to know for your exam, and I have already made your exam, so it's 40 questions. 5 questions are from chapter 1. And about the remaining 35 questions are an even split between chapter 2 and chapter 3. So this lecture, I'm going to record about an hour of it, take a pause, and then I will record the remainder of chapter 2 in a separate lecture. I think that's the only way to do it, is to save 2 separate lectures that will combine into chapter 2 and post them. So, all right. Also, before we get started, I just want to go over some other announcements in regards to. Close that thing here. Whoops. In regards to your term paper. Give me a sec here. All right, so all the information that you need to get started and work on your term paper is posted on my courses. All right, so you basically have 3 due dates for this term paper. The first due date is going to be March 10th. And that's the due date that you're going to choose your topic, right? So also by that date, either you have chosen a topic from this list, or you have settled on a topic with me, okay? So if you don't want to do a topic on this list, if you have another idea, or you see something in the next couple weeks that you're interested in, that's perfectly fine. I just need to approve it. All right, I want to make sure it's a good topic that is broad enough, but specific enough, kind of has that balance so that you'll be able to, you're not going to be either overburdened with information that you need to summarize into a paper, and also that you will have enough information. So that being said, by March 10th, and honestly, you should have this picked out way before then so that you can begin working on it. You need to either choose a topic and post it on this discussion, or post a proposed topic on the discussion for me to let you know if I approve it or not. Okay, so the second thing that is due will be an outline of your paper. That's due on March 16th, and then the final draft of your paper is due March 30th. I believe that's the day before spring break. So it's good you'll have this thing all done behind you before you go on spring break. But that's also midterms week, so just plan appropriately. I don't want you guys having to cram this the night before, and I want you to spend a good time on it and write a good paper. All right, so and both of these, the outline and the final draft, you're going to upload here on Dropbox by the due date. If you look, so here under research paper writing guide, so I go over like the requirements for the final draft, the requirements for the outline, and then also just some general guidelines on what I'm looking for in this paper. Okay, I just want to stress that these are guidelines. Okay, so depending on the topic, some of these things in the guidelines may or may not be applicable. Okay, so you know, how you develop your paper, I think, you know, I'm generally going to be looking for these things, but in the end, they might not all be applicable. All right, please follow all the formatting requirements. Please take note about plagiarism in AI. So AI is prohibited for this paper. It's going to run through Turnitin and the AI detector, and you know, these are pretty good. I've been using these for a very long time, and I will review those results carefully. Okay. Everything about your formatting. And then if you guys have specific questions about this, please reach out. But please start looking this over and start considering the topic that you want to do, you know, pretty early on, so that you can have time to do the research, develop the outline. One other thing I want to mention, you know, I think if I was writing this paper, a majority of the amount of time that I would spend on the paper, in my opinion, is actually writing the outline. I think that's where most of the work comes in. That's where all the research comes in, understanding what you're writing about, organizing, you know, the different points that you're going to bring up, and choosing your sources and all that. You know, after that point, I think putting together the paper, although it's a step, is going to be, you know, relatively straightforward as long as you did a really good job on the outline. Okay. Okay. So again, if there's any questions about that, please reach out. And I'm also here to help you with this paper. I don't like to, like, if you just want to send me a draft before the due date, say, what do you think? I don't typically do that. But if you want to meet over Teams, I will go over, you know, any specific questions that you have. But in general, you guys have a lot of room here with this paper, you know, to kind of go a direction that you want to go, and hopefully write about something that you're interested in. You know, my hopes is that after writing this paper, that knowledge will stay with you for a very long time. Okay. So all that is posted to my courses. All right, let's get going here on chapter 2. Now, just to note about chapter 2, so those of you that are bio majors, biotech majors, and basically anyone here that has taken Biology 1 or even any non-majors intro college level biology course, this should all be review for you. The purpose of this chapter is to get everyone on the same page, because as we move forward and get into all the methodologies that biotechnology encompasses, you're going to need to know most of this basic information to fully understand the methodologies. Right? If you don't understand DNA replication, trying to understand PCR is almost impossible. So, and that's just one example of many. So this is a pretty important chapter. And I would recommend, so in terms of what you're responsible for in the book, so pages 30 to 56, it's a majority of chapter 2. Really the only section that I removed is the very last couple sections. And then I've also included some suggested practice questions for you. So these are found on page 58 and 59. They're relatively simple, I think, but I would recommend that you, at a minimum, answer these questions in preparation for your exam. Just one last thing. So regarding your exam is coming up here. Let me just double check the due date. I don't want to say something wrong. So yeah, exam 1 is due on February 9th. All right, so the way this is going to work, if I can show a calendar on here. Hold on a second. Let's see, calendar. All right. So February 9th is your due date. So that is here. So it's that Sunday. All right. So here we are in this week. So Sunday starts the week. So this is your week to do chapter 2. This is your week to do chapter 3 and to take the exam. Okay. So, 26 here, I'm posting the lecture for chapter 2. And then on the 2nd, I will post the lecture for chapter 3. And then you have that week to review and study chapter 3 and chapters 1 through 3. And then take the exam, which by the 9th. But you can take the exam any day that week. All right. It'll become available. I believe it's on the 3rd at a minute past midnight. And then you have until 11:59 p.m. on Sunday to take the exam. It's 40 questions. And like I said, 5 questions from chapter 1. And then the remaining 35 are pretty much evenly split between chapters 2 and 3. Okay. I would recommend that you answer all of the questions in the back of the book, in the back of the chapter. And don't forget about that resource that you have. Go to my courses, you go to course content. There's a link to some supplemental resources by the publisher, right? Companion website. All right, they have, let's see. Oh, I thought they had answers to the end of chapter questions. Okay, they don't. The chapter quizzes are a good resource here. Also, the essay question flashcards. Even though you're not going to have essay on this exam, it's all multiple choice. Okay, let's get going here at chapter 2. So this is going to be a review of basic cell function, structure and function. We're going to talk about the organelles, the components of the cell. We're going to talk about the molecules of life, right? So macromolecules, carbohydrates, proteins, nucleic acids. We're going to talk about chromosome structure, how DNA is replicated, a little bit about genomes. I did take out a lot of information from that section. We're going to talk about transcription and translation, process of going from DNA to RNA to protein. We're going to look at how genes are regulated. All right, we're going to have the classic lac operon example, and then we're going to talk a little bit about mutations, what things can cause them, how they manifest themselves in terms of the DNA code, and then potential consequences of DNA mutation. All right, so the cell is the most basic unit of life, right? And all the cells on the planet, if you look at them, they all have some things in common. And one of them is that they utilize DNA, right? So it's a universal code among all living things and living cells, all right? So viruses are not cells. They are acellular because viruses depend on the ability to hijack living cellular machinery in order to copy the virus over and over again. All right, so some common structures that all cells have, they have to have a membrane that surrounds them, right? This is not just a simple boundary. Don't think of the plasma membrane as just what separates the inside and the outside of the cell. It does a lot more than that. It's one of the most important structures in a cell. If you think about what defines a cell, it's its gene expression and also what gets in and out of the cell. So the plasma membrane is a big factor in deciding, what gets in and out. It's made of phospholipids. It's really 2 layers that go around the cell. You have an interior of the cell. So the interior is made up basically of 2 things. It's made up of the cytosol. It's spelled Cytosol, which is like the liquid fraction inside of the cell. And then the cytoplasm, which includes the cytosol. And then all of the organelles that are floating around, excluding the nucleus. Right? So the nucleus towards, if you picture like a splattered egg, the nucleus in the center, and it contains its own nucleoplasm. And then the organelles. So organelles are like tiny organs. Think of it like that. They're cellular organs. They're structures that have specific cell functions, specific functions in the cell. So one important thing to distinguish is the difference between cell types. There are basically two families of cells on the planet. Right? You have prokaryotic cells, which in general are bacteria, and you have eukaryotic cells, which are everything else. These make up plants, animals, fungus, um, and another category called protists. But protists are cellular, and they're made up of eukaryotic cells. There are about 15 hallmark differences between prokaryotic and eukaryotic cells. We're just going to cover a few of them in this lecture. Uh, One of the most notable ones is size. All right, so prokaryotic cells are uh, immensely smaller than eukaryotic cells. Right? Bacteria are much smaller than mammalian cells, for example. All right. Also, what prokarya means in its origin, its root name, is no true nucleus. Right. So prokaryotic cells do not have a nucleus in it. Remember, a nucleus is not a requirement to be a cell. Right. You just have to have a membrane, organelles, and cytoplasm. But it still has DNA. It's just not packaged in the nucleus. It's kind of organized through another means. We call that area in bacteria that contains the nucleus the nucleolus. Um, and no membrane-bound organelles um, are present in prokaryotic cells. So eukaryotic cells do have a true nucleus, and they do contain membrane-bound organelles. And it's the presence of that membrane that is kind of unique within the cytoplasm to eukaryotic cells. Right? Prokaryotic cells have a cell membrane, but they don't have membrane structures inside of the cell. Here's a typical bacterium prokaryotic cell. This would be an E. coli. You can see the flagella that it comes off of it, and the nucleoid region um, inside. I think I might have misspoken just a couple seconds ago calling this a nucleolus. It's a nucleoid, I'm sorry. So these are ribosomes right here. It's a structure that's responsible for generating protein in the cell. It's like the protein factory. And, it's basically reading RNA and building protein off of that. But those are structures that are found in all living cells, right? It's an example of an organelle in this cell. One thing bacteria have that some eukaryotes have is a cell wall. Okay, so plant cells also have a cell wall. And the cell wall in bacteria serves as a way to protect the bacteria from blowing up. Right? So bacteria live in diverse environments, right? They can be exposed to, they can live on surfaces where rainwater is falling on top of them. And that creates a condition where the cell starts to fill up with water, right? Because of osmolarity. And it will do that until the cell blows up. So that's called cellular lysis, and it is prevented by this rigid cell wall that it contains. It's made up of a very tough type of sugar called peptidoglycan. The cell wall in plant cells is different, serves a similar purpose, but it has a waxy component. All right, so again, don't be scared at this list. I think when you read through it, a lot of it's going to come back to you from Bio 1, or if you haven't taken Bio 1, possibly high school biology. But these are all the different organelles in a typical eukaryotic cell. All right, so it's split it up over to plant cells and animal cells, and you can see a lot of them are shared between both types. Some of them are unique. Okay. So we're going to go through most of these here. If I don't mention it, it's not on the exam. So, all right. So as I stated earlier, the plasma membrane, a very important structure. It's not just a barrier. It also is it's a dynamic structure, right? It's fluid, and all of the proteins that are embedded in the lipid bilayer kind of can move around in this fluid. It's called like a fluid mosaic model. So mosaic meaning different, meaning you have a lot of different proteins that are embedded in the membrane, and fluid means that they can move, right? So, it has particular roles like cell adhesion, transporting substances in and out of the cell, and whatnot. All right, the cytosol is the interior of the cell, excluding the nucleus. The liquid fraction, it doesn't include necessarily the organelles, even though the organelles are found in that area. It's just the liquid fraction that makes up the cytoplasm. Cytoplasm is cytosol plus organelles. Right? Organelles are specialized function, have specialized functions in the cells. Right? They perform chemical reactions. They make energy. They digest substances. Right? They have hydrolytic enzymes inside of them. So we're going to talk about a bunch of organelles that are found in eukaryotic cells. Right. So this table is just kind of doing another comparison. Some keywords here for plasma membrane. You should understand lipid bilayer, right? It's a double layer of lipids, phospholipids specifically. And there's also some cholesterol there to kind of give the cell some strength. Right? But that's the part that makes the barrier. And then embedded all throughout that membrane, you have proteins that can extend to both the outside and the inside of the cell if they're transporting something in and out. Some are receptors on the surface of the cell that can bind to a ligand and then send a signal in the cell. You have markers on the cell surface that can, for instance, allow the immune system of an organism know that that cell belongs in the body. All right, so I'm going to go through most of these here. I'm just going to kind of give you the short and sweet for what these organelles do. So, so centrioles are found in eukaryotic cells, and they are involved in cell division. Right, they have an important role in cell division, where they produce this spindle. It's called a mitotic spindle that basically pulls chromosomes apart so that the cell can undergo division. It can undergo mitosis. Not all cells are ciliated, and cilia is found in both eukaryotic and prokaryotic cells, but they're these basically short hairs that extend out of the plasma membrane and either can provide movement for the cell or can be used to collect substances and bring it in close proximity to the cell. A flagella is a structure a lot longer than a cilia, and it's used for movement. Okay, so the only mammalian cells that have flagella is the human sperm cell, but it's very common in prokaryotes that's used for movement. Like in our trachea, for example, we have ciliated cells. That cilia is there to kind of trap dust and debris. And also, like when you cough and you expel mucus that's in your lungs, the cilia kind of moves that stuff up out of you. A Golgi apparatus. So Golgi, I like to say think of it like it's the UPS or the FedEx of a cell. It's a processing center. So when proteins get made, the Golgi then takes them in and maybe tags them with some sort of information that will direct that protein where to go if it leaves the cell. Right? So think of it like a processing center or a shipping center of the cell. Golgi is shown right here. This is that's Golgi. Right? Usually it's found in the periphery of the cell. And one end of it is receiving, one end of it is exiting. It's called cis and trans. You don't need to know that. So the cells also have like a skeleton that gives them shape and rigidity. And these are made up of intermediate filaments. Right? So they will provide some physical support to the cell. Cells have to break down substances, digest substances. All right, lysosomes are tiny organelles in the cell that provide those enzymes that break them down. Don't worry too much about microfilaments. Uh, Microtubules are important in cell division, right? So they kind of grow out of the centrioles. They're what grab the chromosomes and pull them to opposite poles. Mitochondria is a very important organelle that produces ATP. Right? ATP stands for adenosine triphosphate, and think of it as cellular energy or like energy currency in a cell. Okay, so whenever an enzyme needs energy, it will get it from a molecule of ATP. Um, if you were to shut off production of ATP in the mitochondria, the cell would be depleted of all the ATP it has in a matter of seconds. Right? So that's why I like to call it energy currency, because it's made very quickly and it's also used very quickly as well. Pretty much everything the cell does in one form will need energy, and this is like the magic molecule and how to get that. So the process of generating that ATP occurs in two places in the cell. It occurs a little bit in the cytoplasm, a process called glycolysis, and in cells that use oxygen, a lot of ATP is made in the mitochondria through a process called oxidative phosphorylation. So mitochondria is a unique organelle because it's double membraned. Like lysosomes just have one membrane. It kind of makes a little vesicle that has digestive enzymes. But the mitochondria and another organelle called the chloroplast that's found in plant cells has two membranes. And the fact that it has two membranes is important in its function because it creates segregated areas within that organelle. And that's important if you need to have different chemistry in different areas to provide whatever function you're doing. That's an important cellular feature. All right, peroxisomes. Very common peroxisome that's found in mammalian cells contains this enzyme catalase. Okay, hydrogen peroxide is a waste product of oxidative phosphorylation, so what occurs in the mitochondria. So as the mitochondria is making ATP, it's also producing this hydrogen peroxide, which as it builds up in the cell can become very toxic, right? It's oxidizing. It can start damaging molecules like proteins and DNA. So peroxisomes have this enzyme catalase, which breaks down the hydrogen peroxide into basically water and oxygen. Ribosomes, very important structures. Remember, they're the site of protein synthesis. Okay, so they're protein factories. They read RNA and they build a protein based on that RNA sequence. So there are there's a membrane that surrounds the nucleus, right? It's called your nuclear membrane, and it actually extends out. If you look, here's your nuclear membrane right here. And you can see right here, it's kind of showing it, how it's extending out right in the periphery of the nucleus. And this is called the endoplasmic reticulum, specifically the rough endoplasmic reticulum, because it's a site where we find a lot of ribosomes, right? You can have ribosomes that are free floating around in the cell, but a lot of them are, they're associated with this ER, as it's abbreviated, or endoplasmic reticulum. And as you can guess, so if the ribosomes are making the protein, the role of the rough ER probably has something to do with the proteins, because they get made right there on the surface of the ER, and then it can come into the ER, and the ER can assist in folding the protein. We're going to talk a little bit about proteins later on and how their function is completely determined by their shape. So if they're not folded correctly, they won't work right. So the ER has an important role in folding those proteins. There's a different type of ER that's also present in the cell called the smooth ER, and it has another role that's more geared towards like lipid formation. So if the cell needs to grow its cell membrane, pieces of the smooth ER can break off and help develop the plasma membrane. This reticuli are membranes, right? They're basically folded up membranes, and the rough ER is actually an extension of that nuclear membrane. Okay. All right, and then you may see vesicles in a cell as well. So this is, a single membrane that surrounds a substance, right? If a protein is getting ready to be exported from the cell, it can't really just go up to the membrane and go out. It has to be wrapped in a vesicle. And when it's wrapped in a vesicle, then it can actually, because the vesicle membrane is made from phospholipids, just like the plasma membrane. When the vesicle gets in contact with the plasma membrane, it can fuse, and then whatever's in the vesicle can leave the cell. All right, all eukaryotes though have a nucleus. It's where the DNA is. It's surrounded by a nuclear membrane, also called the nuclear envelope. It has a kind of a central region called the nucleolus. We'll talk about that later on. But it doesn't have to be the dead center of the cell. Nuclei can think of a cell like a splattered egg, right? It's never dead center. It's a little bit off to the center. All right, so all that DNA in the cell, in the nucleus, if you look at a cell under a microscope, when you look at the nucleus, you can see the DNA in there, but it doesn't look like what you're probably thinking of when I say the word DNA, which is chromosomes. You don't see the chromosomes in there. Instead, you see this speckled appearance that the nucleus has, granular appearance. And this is all the DNA in a very relaxed or diffuse state. Okay? It's not tightly packed like chromosomes are, but it's the same DNA there. What happens is before the cell is about to split and undergo mitosis, all the DNA then starts to really condense, and the chromosomes become visible during that cell division during mitosis. Right? What makes the nuclear envelope unique compared to the plasma membrane is the presence of pores. So there are pores all over the nuclear membrane that allow and traffics certain molecules into and out of the nucleus. Now, all the DNA stays in the nucleus, but as we're going to go over in this chapter, two very essential processes that occur in the cell are transcription and translation. The way information flows in the cell is it goes from DNA to RNA to protein. When you go from DNA to RNA, that's transcription. When you go from RNA to protein, that's translation. Transcription occurs in the nucleus. Okay, so DNA is read, RNA is made, and then the RNA has to leave the nucleus and travel to the ribosome. The ribosome reads the RNA and builds a protein. So those nuclear pores are very important to allow that RNA to exit the nucleus. Plant eukaryotes will have vacuoles. Vacuoles are really large sections of the cell that are typically provide storage, right? So you have starch vacuoles, you have water vacuoles. So the mitochondria in cells, as we talked about, remember that's like the powerhouse, the battery of the cell that produces ATP. I said it's a double membraned organelle. And another organelle that's found in eukaryotes that has two membranes is a chloroplast. The chloroplast in plant cells is where photosynthesis takes place, right? The process of photosynthesis involves generating glucose from just a source of carbon and light energy. And again, it's having those separate compartments within the chloroplast, how it's organized, that permits it to do a very complex function like photosynthesis. All right, so now we're going to talk a little bit about the macromolecules. And the first one is nucleic acids in general. So there's basically two types of nucleic acids, right? You have deoxyribonucleic acids, or DNA, and then you have ribonucleic acids, or RNA. Right? We're going to first talk about DNA, which again stands for deoxyribonucleic acid. The Chargaff A, T, C, and G. So you can disregard that. You don't need to know his specific experiment. But basically, what he discovered was that the DNA molecule is made up of 4 different types of molecules. They're called nucleotides or bases. Sometimes they're called bases. And they are either adenine, abbreviated A, thymine, abbreviated T, cytosine, abbreviated C, and guanine, abbreviated G. All right, so these are the building blocks of DNA, and it makes up a code, right? A, T, G, C, C, G, C, right? It's a string of these nucleotides joined together that make up DNA. So when you look at the structure of a nucleotide, it contains basically 3 things, right? There's a phosphate group, a sugar group, which is made up of deoxyribose, and a nitrogen base, right? So of all 4 nucleotides, the deoxyribose and the phosphate are the same, but the nitrogen base varies. And A has a particular structure of nitrogen base, T has a different one, and so on. Right. So if you look, like here's a generic nucleotide here, right? So here's the deoxyribose sugar, right? 5-carbon sugar, 1, 2, 3, 4, 5. So that number with the prime number indicates the number carbon of that sugar. Here's the phosphate. And then the nitrogenous base is stuck onto that first carbon of deoxyribose. For DNA, here are your 4, 1, cytosine, adenine, guanine, and thymine, your 4 possible nitrogen bases, right? Now, I said DNA is one type of nucleic acid. The other is ribonucleic acid, or RNA. So RNA is very similar to DNA, but one of the differences is that it doesn't use this deoxyribose sugar. Instead, it uses a ribose sugar, which is a little different structure. You certainly don't need to memorize these structures, be able to draw them or anything like that, but you need to know about this and about what we're talking about here. So now, another difference between DNA and RNA, if you look at the nitrogen bases that comprises DNA and RNA, it's a little different. So DNA is A, G, C, and T, and RNA doesn't have T, instead it has U. Okay, if you see here, thymine, which is not present in RNA, whereas uracil is, and thymine's only in DNA, right? So you can start figuring right off the bat, well, a DNA code and an RNA code are very similar. One of the only differences that we've kind of talked about right now is that DNA has T's, RNA has U's. Okay. All right, so here are 3 of the most notable scientists that contributed to determining the structure of DNA. Why is that important? Well, basically, the field of biotechnology really couldn't start without this. Like, you can't manipulate a molecule unless you know the structure of it. And, DNA manipulation is one of the foundational things in biotechnology. So these 3, Rosalind Franklin, James Watson, and Francis Crick, all contributed to the discovery of the structure of DNA. Watson and Crick got all the credit. They published this paper in Nature in 1953. It's considered to be the most perfect scientific publication in history. It's funny, it's only 2 pages long, but it's got it all there. And, Watson and Crick didn't really do any experiments either. They relied off of the data from other scientists to just kind of puzzle and piece together the actual structure of DNA, and then published this work and got all the credit. One individual that whose work was invaluable to the discovery of the structure of DNA is Rosalind Franklin. She did this complex method called X-ray crystallography that showed that the DNA, like the overall shape of the DNA molecule, was a double helix, kind of like a winding staircase. And X-ray crystallography is pretty cool. So, you take a substance that's invisible, like DNA, and then you crystallize it, right? So now you've made that tiny little molecule larger. Still can't see it necessarily, but you can now start detecting it with sensitive equipment. They didn't have the microscopes that would really be able to see those crystals back in the 1950s. Now you can see them quite easily. But after you have the crystallized structure of DNA, then you can bombard it with X-rays. And then how those X-rays bounce off of the crystal, and you detect it with, by surrounding the crystal with X-ray film. And she was able to, she and her lab were able to figure out, how to interpret those X-ray crystallography data into determining structure, and she was able to determine that it's helical structure. She passed away before Watson and Crick were given the Nobel Prize, and Nobel Prizes are not given to individuals that are no longer with us. But a lot of people, suppose that she didn't get the credit that was due because possibly she was a woman. So kind of unknown. All right. So these nucleotides are arranged in a sequence, and they're stuck together by a particular type of covalent bond called a phosphodiester bond. Right. So basically, a carbon from the deoxyribose binds to the phosphate of another nucleotide. That creates a phosphodiester bond. When you look at the DNA molecule, because of the way the nucleotides are arranged, the molecule has polarity. If you imagine like a string of nucleotides, one side of the molecule is termed 5', and then the other side of it is termed 3'. What determines that 5 or 3 is basically the number carbon that is sticking off, like off the butt of the molecule. So it'd be 5' would be this carbon is sticking off on one end, 3' is this carbon is sticking off on another end, right? So this figure can kind of show that a little better. So here's the 3' end of this DNA molecule because the 3' carbon is on this side. Here's the 5' end, right? Because this 5 carbon is right here with the phosphate. DNA is a double helix, right? It has this helical structure, like a winding staircase, and it gives the molecule 2 repeating grooves. So you have a major groove, which is a little larger, and another smaller groove, which is called a minor groove. And these grooves get important later on when we're talking about like transcription factors and things that bind to DNA. DNA is a double-stranded molecule, usually. When it sits naturally in the cell, most often DNA exists as a double-stranded molecule. All of the DNA that's in the nucleus that makes up chromosomes is double-stranded, which means you have one side here that's made up here. We've got 1, 2, 3, 4 nucleotides, and then another side here, which is made up of 4 nucleotides. Now, if you know the sequence of one side here, so here's the sequence C, T, G, A, you can determine the sequence of the opposing side, the complementary side or complementary sequence, because as a rule, C always binds to G and T always binds to A. Okay. Another thing you'll notice, what connects the 2 complementary strands of DNA is not a covalent bond. It's just a weak hydrogen bond. And that's an important component of DNA. The ability for this molecule to actually unzip, for those complementary strands to separate, is important with a lot of uh, biological techniques that are performed on the DNA molecule. Okay. All right. So why is this molecule so important? Well, the DNA and the cells have a lot of it, right? Our cells, we've got 46 chromosomes. Okay? These chromosomes are long stretches of thousands and thousands, hundreds of thousands of nucleotides, right? So you have these massive sequences, and you look at a chromosome, it is divided. It's not divided up. It is organized into genes, right? All along the chromosome lies the genes, which are specific DNA sequences that usually code for proteins, right? So if you think of DNA as the informational molecule, the protein is the functional molecule. Just like there are thousands of genes in the nucleus, there are thousands of different proteins that all have different functions. All right. So, the definition of a gene is funny, though. Most people think of a gene as a sequence that codes for a protein, and most of the time that's true. But really, you should think of it as any DNA sequence that is used to produce an RNA. Okay. Many DNA sequences are transcribed in the nucleus, and RNA is built from them, but that RNA does not become a protein. That RNA in itself is a functional molecule. A lot of techniques in biotechnology exploit this and utilize the RNA that kind of comes off and doesn't become a protein. I'll talk more about that. All right. So, a gene has a lot of information in it. The size of genes varies. Anywhere between 1,000 and 4,000 nucleotides, and there are genes that are a lot smaller than that as well. This is a very average number. Your genes determine your genetic makeup, right? It's the cellular blueprint for the cell, right? So you think of the genes that you inherit from your biological parents, right? So all of those genes collectively determine all of your traits, right? So think of a trait like eye color, right? So, you have genes for eye color on your chromosomes, right? And there exist on different chromosomes, sometimes different versions of those genes, which we call alleles, right? So alleles are like genetic variants of a trait. So eye color would be a trait, and then you could have the allele for blue eyes or the allele for green eyes. All right, chromosomes are made of DNA. They are a single piece of DNA. They contain many genes on them. Different organisms contain different numbers of chromosomes. The number of chromosomes that an organism has nothing to do with the complexity of the organism. A potato has over 100 chromosomes, if I remember correctly, whereas we have 46. Okay, so a lot of that is being sorted out with evolution and has to do with um, content of DNA that's non-coding, but the number of chromosomes don't have anything to do with complexity, right? When you see a chromosome, you know that the DNA is very tightly packaged, right? There are molecules called histones, a type of protein, that package the DNA. DNA is always packaged, but when it's visible as a chromosome, it's very tightly packaged. When it's more loosely packaged, it's called chromatin, like we were saying before. When you're looking at a cell, that's not actively dividing, and you see the nucleus, the DNA looks speckled, and it's chromatin. Right? So yeah, during cell division, chromatin is more tightly packaged into chromosomes. So chromosomes become visible. All right, so where do these chromosomes come from? Well, they come from your parents, right? So you have in all of the cells in your body, except red blood cells, which don't have any chromosomes, and except for sperm or egg cells, all of the other cells in the body, which are called all of the other cells in the body contain 46 chromosomes in them. All right, so 23 of them you get from mom, and 23 of them you get from dad. And then when you look at like all 46 chromosomes, look at the pairs of them. 22 of those pairs, we call them autosomes. And they're basically all the chromosomes excluding the sex chromosomes. So the chromosomes that determine sex are two specific chromosomes that cells have that can either be X or Y. All right, so males have an X and a Y. Females have two X chromosomes. Right, so X and a Y makes a pair. Two X's make a male and also make a pair. So when you pair up the sets of 23, right? You got 23 here, and you pair them together. Let's say if we're counting through 1 through 46, we got chromosome number 1 over here on this side, and we have chromosome number 30 on this side. And those are pairs. They have the same genes on them, but they may have different versions of those genes on them, called alleles. And those pairs of chromosomes we call homologous chromosomes or homologous pairs. Okay, a lot of information. So all of the cells in your body, except red blood cells and sex cells, are diploid because of this. Right? They have essentially two copies of the genome. Right? Ploidy is like a fancy word for genome. And diploid means you have two copies, two sets of 23. All right. When you look at all the cells in the body, all right, you've got the gametes, which are the sex cells, which are not diploid, they're haploid. That's why, like a sperm, which is haploid, and an egg, which is haploid, come together during fertilization and produce a zygote, which is diploid. 23 plus 23 equals 46. So what type of a cell is a kidney cell? Well, it's a somatic cell. It's a diploid cell. It has 46 chromosomes in it. All right. So looking a little more at chromosome structure. Right. So before a cell has to divide, right, before a cell divides, you have one cell that's going to now split into two cells. And each of those two daughter cells have to have the same genes, have to have the exact same copy of that parental cell. All 46 chromosomes, an exact copy of all the DNA, hopefully no mistakes. So before the before the cell splits, the DNA gets copied, right? So those 46 chromosomes basically produce next to them, you will have a copy of that chromosome. We call those copies chromatids or sister chromatids, right? Results from DNA replication, and it occurs before the cell splits into two. All right, so when you look at chromosomes, with their attached chromatid, their attached copy, there is like a region. There's a photo here of it. Yes. Okay. So this is one chromosome, right? But there are two chromatids that make it up, right? So here's one chromosome right here, and here's its chromatid, right? And together we would call these sister chromatids. So you can see how it's tightly packed here. There's a visible chromosome. This is actively during mitosis. So this is when chromosomes are visible, and this is when the DNA is tightly packaged. And it's because of these histone proteins right here that enable this coiling and packaging of the DNA. These are long pieces of DNA. You got to organize it, you got to package it, you got to fit it all in the nucleus of the cell. And it's because of these histones that you can do that. So here's that centromere. It's what connects sister chromatids together. And it kind of gives, one side of the chromatid could be long, the other could be short. and they're labeled P and Q, the arms of the chromatids. And the very ends of chromatids are called telomeres. All right. So what we're going to learn when we cover DNA replication is that because of the way the process is set up, the very ends of pieces of DNA will actually start to degrade over time. They'll get shorter. The DNA molecule will get slightly shorter and shorter every time the DNA gets copied. And it's that telomere region that is undergoing that. And so there are enzymes, enzyme called telomerase that works to kind of replace that degrading amount of DNA. And the telomere is a certain structure that kind of protects the ends of the DNA from that process. All right, so here you're seeing 46 chromosomes, okay? And obviously this is during mitosis, and this is a common type of biological method called producing a karyotype, right? Which kind of displays all of the chromosomes. Here's your 22 autosomes, or 22 pairs of autosomes. Here's your 2 sex chromosomes. So this person is a male because it has an X and a Y. And this right here, each of these sets would be a set of homologous pairs, homologous chromosomes. Now, what you don't see here are the sister chromatids, right? So that's an important thing to distinguish. What you're seeing here are homologous pairs. So karyotypes are done because you can basically stain the chromosomes looking for specific DNA sequences. And it can be used to just visualize, like all the chromosomes. If a chromosome was broken significantly, we could see it with the karyotype. If the cells had 3 copies of a chromosome, we could see that on karyotype. The syndrome trisomy 21, right, which causes Down syndrome, characterized by having 3 chromosomes for number 21. So karyotypes are often accompanied by another technique called fluorescent in situ hybridization. There's not a picture of it, but fluorescent fish or fluorescent in situ hybridization uses DNA probes, right? So DNA probes are like short oligonucleotide sequences, right? Small, maybe 20 base pairs long, and they have conjugated to them a fluorescent molecule, right? Now remember, DNA is complementary, right? So one strand binds to another. So if you take that fluorescent short piece of DNA and you put it in the cell, and as long as the sequence is complementary to a sequence somewhere here in the genome, it will find and bind to it and then glow, as long as the conditions are right and you're putting the right light on it and whatnot. So that can be used to find specific sequences, in a karyotype or in a living cell even. There's a lot of applications for fish, and it's one of your topics in one of your suggested topics for your paper. Okay, so a very important process occurs before a cell divides. Right? Remember, a diploid cell undergoes mitosis and it splits into 2 diploid cells. So how do you get from one diploid to 2 diploid? The only way to do that is if all the DNA in that cell gets copied. Right? And DNA replication is the process by which all of the DNA gets copied. It's basically the most essential step leading up to mitosis, which is active division of the cell. Right? So you think of all of your somatic cells in your body, right, which are all of the cells excluding the sex cells and their blood cells. And those somatic cells, your tissues have to regenerate, they have to heal, or if you're a young organism, you have to grow and develop, right? So mitosis enables growth and repair um, and whatnot. The process to generate gametes or the sex cells is a little different than mitosis, and it's that process we call meiosis. It's very similar to mitosis, but there are some key differences. One of the biggest differences is that it's divided up into two stages. You have meiosis 1 and meiosis 2. And each stage of meiosis, 1 or 2, is like almost all the steps of mitosis. Okay? Some of the stages that might ring a bell: prophase, metaphase, anaphase, and telophase. Those are the steps of mitosis, but they're also the steps of meiosis 1, and then the steps of meiosis 2 as well. So what is the difference? Well, in summary, during meiosis 1, homologous pairs separate into those two new cells that get produced during meiosis 1. And during meiosis 2, sister chromatids separate, and now you have 4 cells that came from one parent cell. This is meiosis, the development of sex cells. And now those 4 daughter cells can develop into either a sperm or an egg. So meiosis is very crucial, though, to genetic diversity, right? So the reason why you are not like an exact replica of your siblings, and you are not exact replicas of your biological parents, is because the meiosis that occurred to generate the sperm and egg that made you had a very important process that occurred during meiosis 1 called crossing over. Picture 2 homologous chromosomes next to each other, and arms of each chromosome will literally cross over, fuse, and then basically switch from one chromosome to another. That mixes up all the genes, and it creates a lot of diversity. And there are other things that produce diversity as well. Which sperm finds the egg, obviously which partner, a mate that you find. But this crossing over event is very important. Also, how the chromosomes line up in the middle of the cell during metaphase and then separate, they do so in a very random fashion. Okay, so they randomly line up in the cell, and they randomly segregate from one side to the other. This is only in meiosis, not in mitosis. All right, so mitosis is a separation of sister chromatids. You're going from diploid to diploid. Meiosis, divided in 2 stages, where stage 1, you separate the homologous pairs, and then in stage 2, you separate the sister chromatids. But in order to do either mitosis or meiosis, you have to replicate all the DNA first. It's the first step before mitosis or meiosis occurs. All right, so you know that DNA is a double-stranded molecule, right? It can be unzipped easily because of those weak hydrogen bonds that are holding the complementary nucleotides together. Remember, A to T, C to G. So there were some very elegant experiments using radio-labeled DNA that discovered that DNA is replicated in a semi-conservative fashion. So it's called semi-conservative replication. Basically, the DNA can unzip, and then a new piece of DNA can be made from each original or parental strand by new nucleotides coming in and binding in a complementary fashion to the parental strand of DNA. So you've got one parent strand of DNA, it separates, and then you have your new nucleotides coming into each strand, and then now you have 2 new pieces of double-stranded DNA from that one piece, that one original strand. The sequence gets copied entirely, and as long as the wrong nucleotide doesn't come in and bind, it's going to be an exact sequential copy of the original parental strand. All right, so One thing I want you to notice here before I get to the next slide, though, because this is important about DNA replication, is you can see on this strand here, the nucleotides are coming in on this side of the new piece of DNA. And on this strand here, the nucleotides are coming on this side of the new strand of DNA. It's an important point because the way the enzyme that performs DNA replication, called DNA polymerase, the way that it functions, it can only move in one certain direction along the DNA molecule. All right, so let's go through the steps of replication. After we get to replication, I'm going to pause the video and then we'll do the rest of the chapter. But so the first step, you got double-stranded DNA. You have to unzip it, right? There's this all-important magical enzyme which unzips the DNA. Right? If anyone ever asks you if you could be any enzyme in the world, what would you be? You say DNA helicase, because then you could unzip their genes. Funny joke. Anyways, okay, so helicase unzips the DNA. It breaks the hydrogen bonds. All right, now DNA polymerase is the enzyme that will build the new strand of DNA, but it can't just arbitrarily bind to DNA and start. It doesn't work like that. The position it will start from is a position that contains a short primer. Right? So after the DNA gets unzipped, there's an enzyme called primase that will lay down a short nucleotide sequence that is actually made up of RNA, not DNA, and it's called an RNA primer. So like in this figure here, the primer was on this end of this new piece of DNA. And on this side over here, the primer was on this end of this new piece of DNA. Then polymerase comes and binds to the primer, and it starts moving in this direction down this strand of DNA to build new DNA. What I'll do is I will post to see this mechanism. It can be really helpful to see a video, a cartoon video that shows you the steps, and I will post a video of DNA replication to kind of assist you in if this is difficult to understand just looking at these not very good photos or images. Okay, so helicase unzips. One thing here, so after it unzips, because those hydrogen bonds are broken, the two strands of DNA are not very stable, right? They want to come back together. So there are proteins that kind of line along the back of the DNA strands, stabilize it, and keep the strands from re-annealing. These are called single-stranded binding proteins, also abbreviated SSBPs. And then also, as you're looking down a chromosome, right? So eukaryotic cells have linear chromosomes. I don't know if I mentioned this before, but prokaryotic cells have circular chromosomes. We'll talk about that later on. But anyways, along these linear chromosomes, replication is going to start in multiple areas. These are called origins of replication. So they actually, imagine this is your double-stranded DNA, where a origin is it bubbles, right? So the DNA, the helicase comes in, it unzips it, and it forms what's called a replication bubble. And in order to, be able to synthesize a long piece of DNA like a chromosome, you need to have multiple origins. If DNA polymerase started on one end and went all the way to the other, it would take forever to do, and it wouldn't be very efficient. So, all right, so helicase, single-stranded binding proteins, primase comes in and lays an RNA primer on both parental strands that have been unzipped. Okay, now this is a very easy molecular process. The only thing that students will get tripped up on is this part. All right, so the enzyme DNA polymerase can only travel along the parental strand in one direction. Okay, it can only build DNA in a certain way. It can only add nucleotides to the 3' end of the growing strand. Okay, you have to memorize that. DNA polymerase can only add nucleotides to the 3' end of the growing strand of DNA. So DNA, it binds to the primer, and then it starts traveling down the new strand of DNA, adding nucleotides. Traveling down the parent strand of DNA, adding nucleotides to the 3' end of the new strand of DNA. All right, so this next figure will kind of show that. All right, so here's your double-stranded DNA here. Remember, we had DNA has polarity, 5' on one end, 3' on the other. And the complement piece of DNA has, complementary polarity, 5' to 3'. So it's 5 here, it's 3 here. If it's 3 here, and I'm looking at this 3, then it's 5 here. Okay. DNA helicase is right here, and it's unzipping the DNA. Okay. So DNA helicase is moving in this direction, traveling down the DNA molecule like this, and it's unzipping it as it goes down. RNA primase is going to come in and lay a primer. All right, so it's going to lay a primer here. Now you look at the polarity of this strand of DNA. So if the primer is here and DNA polymerase binds here to this primer, it can move in this direction continuously behind helicase as helicase is unzipping the molecule. And it can do that with no problem, because if this is the 3' end, this is the 5' end of the new growing strand. And remember, it's adding nucleotides to the 3' end of the growing strand, which is right here. It's the blue arrow tip. All right, so this side of the DNA molecule, when it's undergoing replication, it's said to undergo continuously. It's also called the leading strand because of that. Now, the other strand of DNA, as you can imagine, is going to encounter a problem, right? Because when DNA polymerase comes down and binds to the RNA primer, okay, here's a primer, it can only move in the opposite direction, because DNA polymerase can only add nucleotides to the 3' end. So DNA polymerase is moving this way here, but DNA polymerase is moving this way here. So if it's moving this way, since helicase is moving that way, you're going to get gaps in the DNA that wouldn't get replicated. All right. So to overcome this, what happens is this strand is made in segments or fragments that we call Okazaki fragments. All right. Here's an Okazaki fragment. Here's an Okazaki fragment. And they're defined by their RNA primers. Like this RNA primer goes here, DNA polymerase binds, it moves that way, and that's one Okazaki fragment. Primase puts a primer down here, DNA polymerase binds, and synthesizes DNA in this direction, all the way up to that other primer. There's another Okazaki fragment. And it keeps doing that as helicase is going that direction and unzipping the DNA. Okay, so because this is made in fragments, it's said to be the lagging strand, right? Whereas this is the leading strand that's being continuously replicated. This is the lagging strand that's being replicated in fragments. Now, when DNA replication is complete, when polymerase has made all of the fragments, you've got to get rid of that RNA and replace it with DNA, that's making up the primers. On the leading strand, you just have one primer. On the lagging strand, you have multiple primers. And basically, there's an additional type of polymerase that will come in and remove that RNA primer and replace it with DNA. All right. And then importantly, you have to create, this is a series of nucleotides with phosphodiester bonds. You have to join those Okazaki fragments together. You have to create a phosphodiester bond between Okazaki fragments. And there's an enzyme called DNA ligase that does that. Right? DNA ligase joins the Okazaki fragments together. Right? But why is this stuff important in biotechnology? Well, the process of PCR basically exploits all of this mechanism. And the process of sequencing, the processes of RT-PCR and microarrays, you have to have knowledge of the DNA molecule and how it works in order really to understand um, those different methodologies. All right, I just want to see, yeah, so I think I covered everything here, leading and lagging, Okazaki fragments, DNA ligase joining the Okazaki fragments together. RNA primers are replaced with the DNA using it's a different type of DNA polymerase. Right? We're not going to worry about all the different types, but it's like this DNA polymerase that's doing most of the work. All right, I'm gonna. I don't think I guess I need to get a drink, so I'm gonna see if I can just pause this. I can stop recording, and then I'm gonna come back and hopefully I can just post this whole thing as one continuous video, and I don't have to do it, but I might have to post it as two videos. So I will be back here in a little bit. Stop sharing. Stop recording.